My name is Jakayla Levison, and this is my submission for homework 2. My question came from chapter 4, number 4, and it states, eukaryotic organelles may be targeted by environmental toxins or microbial pathogens. Describe 2 examples of disease in humans which results from damage or malfunction to an organelle or other intracellular structures. First, I want to discuss eukaryotic cells. It includes plants, animals, protists, and fungi. When compared to a prokaryotic cell, they are larger, they contain a nucleus, are sexually reproductive, multicellular, contains multiple chromosomes, and has organelles. So, an organelle is a specialized subunit, usually within a cell, that has a specific function. We have the Golgi apparatus, the flagellum, the cytoskeleton, the centrosome, limosome, nucleus, rough ER, and ribosomes, soft ER. We have the mitochondria, we have the peroxisome, we have the plasma membrane, also as the glycolyx. So, today I will be focusing on the mitochondrial organelle and how it can lead to diabetes, cancer, apoptosis, Parkinson's disease. So the function of mitochondrion in the cell, it provides the majority of ATP, which is what we use as a source of energy. And a lot of people think that the nucleus contains all of the DNA within a cell, but that is not true. Each mitochondrion has its own DNA and genomes, and you can have up to 10 to 20 mitochondria in one single cell, and each of those have their own DNA. And since there's so many, and we can have mutations and we can have problems within them. So the topic is mitochondrial dynamics. We have fusion and fission. For fusion, it's fused mitochondria. It increases ATP, mitochondrial mass, and mtDNA mixing. Fission produces fissioned mitochondria. It has decreased ATP, increased number of mitochondria, and mtDNA segregation. This is very important. The balance of fusion and fission is very important. If these are not balanced, it can lead to mitochondrial dysfunction. Mitochondrial dysfunction is a condition characterized by loss of membrane potential to decrease ATP production, decreased respiration or oxidative phosphorylation, leading to a metabolic shift to the glycolysis-dependent ATP generation that takes place outside of the mitochondria, which increases the formation of mitochondrial reactive oxygen species, ROS. Uncontrolled production of ROS can further damage or distract the mitochondrial membrane and its major constituents. It can affect the DNA, lipids, and proteins. Initially, mitochondrial dysfunction can be controlled by fusion and fission activities. That's why this balance is so important. Fusion delays the onset of apoptosis by inhibiting mitochondrial fragmentation, while fission has a positive role on apoptosis. However, the failure of quality control can contribute to the development of degenerative diseases like type 2 diabetes, cancer, cardiovascular disorders, Parkinson's, Alzheimer's, and age-related disorders. Now up here, we have mtDNA. So this is a nDNA and MTA abnormality, so a damage or mutation right here. It could also give you cell stress, which is environmental, Project Asbesto, Scylla, drug, chemical, or virus. These can lead to any of these. So this is where damage or mutation, because it could damage the DNA, it could damage your proteins. So when you have damage or mutation, that leads to mitochondrial dysfunction. As I said before, Fusion and fission can help stop this, but if something bad happens, if it's not balanced, it can lead to a whole variety of things. First, we have metabolic dysfunction, which can lead to, diabetes, obesity. We have cancer, which leads to the Werberry effect in cancer. It can give you abnormal inflammation and immunity. It can also affect your aging and apoptosis.